welcome to our video on isomers. In this video, we're going to talk a little bit about what isomers are and look at some important examples of them in biology. Isomers are molecules that have the same molecular formula, so they have the same number of the same types of atoms, but the atoms are arranged differently. As we've seen in other settings, in biology there's a strong correlation between structure and function. So if we change the structure of the molecule, we can alter its properties. It's important to note before we go on, not to confuse isomers with isotopes. Remember that isotopes are atoms of the same element which differ in their number of neutrons. In the case of isomers, we have the same number of everything, but we put it together differently. The first type of isomer that we're going to look at are structural isomers. There are four main ways that we can build structural isomers. The first of which is having either a straight chain, as demonstrated in the left-hand molecule, or a branch chain, which you can see on the right. Both of these molecules are C5H12, but in one of them we have all of the carbons in a line, and in the other we have one of the carbons forming a branch on the side of the molecule. Our second type of structural isomers are chains versus rings. We'll see this a little more when we come to look at glucose and the other carbohydrates in our section on macromolecules. But certain types of molecules can exist in these two forms, and can even switch between them depending on the conditions. The third way we can have structural isomers is to have double bonds in different positions. In the example here, we're revisiting our 5-carbon molecule from before, but now we only have 10 hydrogens instead of 12, because 2 of the carbons are double bonded to each other, giving them each one less valence electron to share with other atoms. In the molecule on the left, we have the double bond between carbons 1 and 2, while in the molecule on the right, it's between carbons 2 and 3, which will alter how the molecule behaves. Our final type of structural isomer is one in which we change the location of certain functional groups of atoms that are typically responsible for the chemical reactions of a given molecule. Now, before I tell you the groups that have moved, I'd like you to pause the video for a moment and see if you can find them. And the difference between these molecules really comes down to the location of a single hydrogen atom. But what that amounts to is one having a carbonyl group on the end with a hydroxyl group on the next carbon, while the other has a hydroxyl group on the end and a carbonyl on the second carbon. This might seem like a really small difference, but it can alter important factors, like how well this molecule reacts or interacts with other molecules. And as we discussed in the very first section, interactions between components are critical for making sure the system as a whole functions correctly. Our other types of isomers have fewer permutations. Cis-trans or geometric isomers are similar to structural isomers, but they differ in the arrangement of groups, specifically around an inflexible structure, typically a double bond. Now, sometimes we get lazy when drawing molecules, and so we sometimes skip writing out all of the carbons. When you see a molecule drawn like this, each point where the lines intersect is a carbon. If the drawing doesn't specify what's attached to that carbon, you can assume that it has enough hydrogen to complete its valence shell. In this case, you can see the 2 lines joining those points that we just identified as carbon atoms, which tells us that there's a double bond here. In this example, the groups in question are the 2 methyl CH3 groups, which are attached to those double bonded carbons. In the cis isomer, the methyl groups are on the same side of the double bond, while in the trans isomer, they're on opposite sides of the bond. Now, this only matters when the carbons that those groups are attached to are joined by a double bond or other inflexible structure. A single bond has freedom of rotation, and so if the carbons are joined by a single bond, the groups can move around that more. Now, the final type of isomer that we're going to look at are enantiomers, and these are molecules that are mirror images of each other. Take a look at your right and left hands for a moment. Can you rotate one to match the other? You can have your thumbs on the same side, but then one hand is palm up and the other is palm down. You can have your palms facing the same direction, but now your thumbs are on opposite sides. This is how enantiomers work. The groups attached to the central carbon vary in their locations in three-dimensional space, so that one molecule is the mirror image of the other. In biology, enantiomers are important pharmaceutically, as many common drugs like ibuprofen or albuterol, for example, exist as enantiomers, but typically only one of the two is active. It's even true of naturally occurring biological molecules, like the amino acid alanine, which exists as two enantiomers, but only one is used to build proteins. Even glucose can be found as two enantiomers, although only one is typically produced by photosynthesis, and the other is rarely seen naturally. Thanks for watching, and please make sure to check out the video on functional groups.